All right, just keep your eyes focused on the light for me. The cranial nerves appear to be functioning normally at this time.
The clinical beginning, a brain that looks normal. The patient sits upright in a neurology consultation room. Motor strength is intact. Cranial nerves are normal. Speech is fluent. MRI shows no tumor, no hemorrhage, no infarct. Cortical volume appears preserved. White matter signal is nonspecific. Blood tests are within reference range. Cerebrospinal fluid shows no inflammatory markers. No biomarker threshold has been crossed. By every conventional measure, the brain appears structurally stable. Yet functional decline has already begun. Neurodegeneration does not begin when a diagnosis is assigned. It begins when neurons lose metabolic efficiency, when white matter conduction slows by milliseconds, when protein clearance systems falter below detection thresholds. Structural collapse is the end of a process, not the beginning. Long before atrophy appears, networks can desynchronize. Long before plaques become measurable, synapses can weaken. The symptoms are subtle but progressive. Tasks that once required no effort now require sequencing. Fatigue accumulates despite normal imaging. The nervous system may be losing timing precision and metabolic resilience, even while scans appear unchanged. The absence of a lesion creates ambiguity. Neurology is trained to localize dysfunction. Stroke produces focal deficits. Tumor produces mass effect. Multiple sclerosis produces plaques. Alzheimer's disease produces medial temporal atrophy and biomarker shifts. In this case, localization fails. There is no clear territory of destruction. There is only decline without anchor. Early evaluations often attribute these symptoms to stress, sleep disruption, anxiety, or depression. Psychiatric framing becomes convenient because the neurological exam is largely reassuring. Insight remains preserved. Memory testing may be normal in structured conditions. Reaction time is slightly slowed, but not pathologic. Imaging does not compel escalation. What defines this beginning is not the presence of catastrophic findings. It is the absence of them. No amyloid positivity above diagnostic threshold. No tau pattern consistent with a named taupathy. No Lewy body signature. No inflammatory cascade. No genetic mutation with established phenotype. The brain appears structurally intact by conventional standards. But conventional standards rely on thresholds. Disease classification in neurology depends on pattern recognition and biomarker density. Below that boundary, it remains undefined. The patient inhabits this space, not healthy, but not diagnosable. At this stage, a degenerative process may already be active at the cellular level. Neurodegeneration does not wait for classification. Cellular stress can accumulate without producing macroscopic lesions. White matter conduction can degrade without producing discrete plaques. Mitochondrial inefficiency can reduce neuronal output without killing the cell outright. Networks can desynchronize without leaving a visible scar. The clinical setting reinforces the illusion of stability. The patient walks independently. Orientation is intact. There is no seizure, no paralysis, no dramatic cognitive collapse. Monitoring is recommended. Follow-up is scheduled. Reassurance is offered cautiously because no alternative can be confidently named. The problem is not that nothing is happening. The problem is that what is happening does not fit existing categories. The nervous system may be experiencing early loss of timing precision, metabolic resilience, and conduction efficiency. But these changes are diffuse, subthreshold, and below detection limits of routine imaging and biomarker analysis. Neurodegeneration without diagnosis begins here, in a brain that appears normal by current standards while function declines steadily. The absence of a name does not equal absence of structural failure. It marks the beginning of a gap between biology and classification. What a diagnosis requires, why naming a disease requires thresholds. Neurological diagnosis is not built on subtlety. A disease is named when structural, biochemical, or clinical features cross defined boundaries that align with established patterns. Multiple sclerosis requires demyelinating lesions disseminated in space and time. Parkinson's disease requires bradykinesia plus dopaminergic response or characteristic imaging changes. Without these thresholds, classification stalls. This framework protects accuracy, but it creates blind zones. Biomarkers are binary by necessity. Amyloid is either positive or negative based on a reference range. Tau is either elevated or not. Autoimmune antibodies are either detected or absent. Yet biological processes are continuous. Protein misfolding begins long before plaques are dense enough to measure. Demyelination may occur diffusely without forming discrete lesions. Mitochondrial dysfunction may reduce neuronal efficiency without causing visible atrophy. Imaging operates under similar constraints. MRI resolution is measured in millimeters. Each voxel represents millions of neurons and glial cells. A brain can lose synchrony and conduction precision without showing a mass, hemorrhage, or overt atrophy. The absence of a visible lesion does not equate to structural integrity at the cellular level. Clinical criteria also depend on pattern recognition. Diseases are defined by clusters of signs that reliably co-occur. A patient must fit the pattern closely enough to qualify. But what happens when decline follows a trajectory that does not align with a named syndrome? Slowed executive function without memory loss. Gait inefficiency without clear cerebellar ataxia. Cognitive fatigue without psychiatric markers. These features resist categorization because they overlap across disorders without reaching diagnostic specificity. Genetic testing adds another layer of limitation. Known mutations account for defined syndromes. Yet most neurodegenerative conditions are polygenic or sporadic. A negative genetic panel does not rule out structural vulnerability. It simply means the known mutations are absent. The absence of a mutation is not proof of resilience. Autoimmune and inflammatory markers function similarly. If antibodies are not detected and cerebrospinal fluid lacks pleocytosis, inflammatory disease is deprioritized. However, neuroinflammation can occur at levels insufficient to trigger standard thresholds. Microglial activation may persist below measurable limits, contributing to chronic degeneration without meeting criteria for autoimmune encephalitis. The consequence of threshold-based diagnosis is that patients below those lines exist in a diagnostic vacuum. They are monitored but unnamed. Follow-up scans are compared to prior studies that were already labeled normal. Biomarker tests are repeated but remain within reference ranges. The disease progresses, but it does so beneath categorical recognition. Medicine requires categories for treatment pathways, research design, and communication. Insurance systems require codes. Clinical trials require inclusion criteria. A condition without a label struggles to access intervention, not because it is benign, but because it is undefined. This structural reliance on thresholds means that neurodegeneration can advance silently until it coalesces into a recognizable pattern. By the time protein deposition is dense enough to measure or atrophy becomes visible, years of subclinical decline may have already occurred. Early inefficiency is dismissed because it does not map cleanly onto established frameworks. Neurodegeneration without diagnosis therefore exists not because pathology is absent, but because classification systems demand density, pattern, and reproducibility. Biology does not wait for classification. It accumulates gradually, diffusely, and variably. The nervous system can degrade below detection thresholds for years before crossing the boundary that grants it a name. The absence of a label is not proof of normalcy. It reflects the limits of detection and the necessity of thresholds. In that gap between measurable pathology and lived decline, unclassified brain failure progresses quietly, unrecognized but real. White matter failure without a name, conduction before collapse. Not all neurodegeneration begins in gray matter. Not all decline begins with memory loss. In many unclassified cases, the earliest failure occurs in white matter, the connective infrastructure that allows distant brain regions to synchronize their activity. White matter is composed of long axonal tracts insulated by myelin, produced and maintained by oligodendrocytes within the central nervous system. These tracts enable rapid signal transmission across the cortex, between hemispheres, and down into subcortical structures. When white matter begins to fail diffusely, function slows before it collapses. Myelin is not static insulation. It requires continuous metabolic maintenance. Lipid turnover, protein repair, and energy supply must remain intact for conduction to remain precise. Subclinical oligodendrocyte stress from age, metabolic strain, vascular inefficiency, or low-grade inflammation can weaken myelin integrity without producing discrete plaques. Signal conduction may become fractionally slower under diffuse myelin stress. Action potentials arrive slightly out of phase. Networks lose timing precision. This desynchronization does not produce paralysis or aphasia immediately. It produces inefficiency. Executive function becomes effortful because frontal subcortical circuits depend on rapid conduction across long association fibers. Task initiation falters not because the plan is absent, but because the signal coordinating that plan arrives late. Multi-step actions require conscious control because automatic integration has degraded. Diffuse white matter failure may not meet criteria for a defined demyelinating or leukodystrophic syndrome. There are no focal lesions. No symmetric hyperintense pattern severe enough to classify. Radiology reports may describe nonspecific white matter changes or even appear normal. Yet microstructural degradation can exist beneath imaging resolution, particularly when damage is distributed evenly rather than concentrated. Interhemispheric communication through the corpus callosum becomes subtly inefficient. Tasks requiring bilateral coordination slow. Cognitive flexibility declines. Switching between concepts or activities requires disproportionate effort. The patient describes mental fatigue rather than memory loss. Neuropsychological testing may show slowed processing speed with preserved recall, a profile that does not align with classic cortical dementia. Axonal conduction depends not only on myelin integrity, but also on energy supply. Mitochondria distributed along axons generate ATP required to maintain ionic gradients. If metabolic stress reduces ATP availability, conduction becomes unstable. Sodium channels redistribute along demyelinated segments. Energy demand increases. The system becomes fragile. Yet unless axonal degeneration reaches a threshold, imaging will not show tract loss. Low-grade microglial activation may compound this process under sustained stress. Even without overt inflammation, low-grade activation releases cytokines and reactive oxygen species that stress oligodendrocytes. The white matter environment becomes hostile to maintenance, but not inflammatory enough to be detected in cerebrospinal fluid. Damage accumulates diffusely. The critical feature of white matter failure without a name is distribution. Because degeneration is widespread and symmetric, there is no focal deficit to localize, no sensory level, no unilateral weakness. Instead, there is global inefficiency across multiple domains. Each network loses timing precision incrementally. This pattern delays recognition because it lacks trauma. There is no seizure, no acute collapse, no dramatic MRI finding. There is only a brain working harder to achieve the same output. Fatigue increases. Errors accumulate. Executive control requires conscious override. The patient senses decline, but the nervous system has not yet crossed the boundary into visible structural loss. White matter failure precedes collapse. Conduction desynchronizes before axons die. The system remains anatomically present, but functionally compromised. Until degeneration becomes dense enough to cause atrophy or gliosis, it exists in a diagnostic blind zone. Neurodegeneration without diagnosis often begins here, in the slow erosion of timing and conduction rather than in overt cell death. The architecture is intact enough to evade classification, yet unstable enough to alter daily function. The brain appears structurally preserved, but its communication network is losing coherence beneath the threshold of recognition. Proteinopathy below detection, tau TDP-43, Alpha-Synuclein in subclinical ranges. Neurodegenerative diseases are often named after the proteins that accumulate within the brain. Amyloid and tau in Alzheimer's disease, alpha-synuclein in Parkinson's disease and Lewy body disorders, TDP-43 in certain frontotemporal dementias, and ALS. These proteinopathies are identified when aggregates become dense enough, regionally specific enough, and reproducible enough to define a syndrome. But protein misfolding does not begin at the moment it becomes measurable. It begins long before detection thresholds are crossed. Proteins such as tau and alpha-synuclein normally exist in dynamic equilibrium. They fold, unfold, interact, and degrade continuously. With age, metabolic stress, or subtle genetic vulnerability, folding fidelity decreases. Small numbers of misfolded proteins escape clearance systems. Under ideal conditions, proteasomes and autophagy pathways remove them efficiently. When clearance slows, even slightly, accumulation begins. Below diagnostic thresholds, these aggregates are sparse and diffusely distributed. They may not form the dense plaques or inclusions typically required for pathological definition of a specific syndrome. They exist as oligomers and microaggregates, disrupting intracellular transport and synaptic stability without producing regionally specific atrophy. The nervous system compensates, but performance efficiency declines. Mixed proteinopathies may occur at low density. A small burden of tau aggregation, combined with scattered alpha-synuclein inclusions and TDP-43, mislocalization may not fit any single disease pattern. Each protein burden may contribute modestly to cellular stress in susceptible regions. Together, they destabilize networks. No single pathology dominates enough to be named. Age-related vulnerability amplifies this effect. Proteostasis, the cellular system that regulates protein folding and degradation, becomes less efficient with aging. Endoplasmic reticulum stress increases. Mitochondrial output declines. Oxidative stress damages proteins at a rate slightly faster than repair systems can manage. The brain may tolerate these changes for years before structural damage becomes visible. Network vulnerability is not uniform. Regions with high metabolic demand, such as frontal association cortex and hippocampal circuits, are particularly sensitive to even low-level protein aggregation. Timing precision degrades first. Executive dysfunction appears before memory collapse. The patient may experience cognitive slowing without meeting criteria for Alzheimer's disease because tau burden is insufficient to produce the classical medial temporal atrophy pattern. TDP-43 pathology illustrates this ambiguity. Small cytoplasmic mislocalizations may impair RNA processing and synaptic function without producing the profound motor neuron loss seen in ALS or the behavioral changes of frontotemporal dementia. The pathology exists, but it does not align with a recognized phenotype. Alpha-synuclein behaves similarly. Low-density cortical deposition may destabilize attention. Networks without producing the motor phenotype of Parkinson's disease or the hallucinations typical of dementia with Lewy bodies. The disease is present at a molecular level, but not at a categorical level. Imaging cannot detect these early aggregates. Standard MRI reflects macroscopic structure. Even advanced PET imaging requires density above a measurable threshold. Below that, proteinopathy remains invisible to routine diagnostics. The consequence is functional degradation without a label. Synapses weaken. Axonal transport slows. Network synchrony falters. Yet cerebrospinal fluid biomarkers remain within reference ranges. No pattern of deposition reaches the specificity required for diagnosis. Pertinopathy below detection can represent a transitional, biological state between health and a defined neurodegenerative syndrome. It is biologically active, but diagnostically silent. It may never consolidate into a single recognizable syndrome. Instead, it may produce diffuse inefficiency that progresses slowly until axonal degeneration begins. Neurodegeneration without diagnosis can therefore arise from cumulative subthreshold protein stress. The brain's clearance systems are overwhelmed incrementally rather than catastrophically. Aggregates accumulate diffusely rather than regionally. The pathology exists, but it lacks the density and pattern required to earn a name. This stage is not benign. It is preclinical in density, not in effect. The absence of a diagnostic signature does not indicate absence of molecular disruption. It reflects the gap between measurable pathology and biological reality, a gap where decline can proceed quietly beneath the limits of detection. Mitochondrial decline and metabolic failure energy collapse without structural lesion. Neurons may fail through mechanisms that extend beyond protein aggregation or myelin degradation. They become vulnerable when energy supply persistently falls short of metabolic demand. The brain consumes a disproportionate share of the body's metabolic output, and neuronal function depends on continuous ATP production. Mitochondria are the engines of that production. When mitochondrial efficiency declines, neural systems weaken before any cell dies. Mitochondrial dysfunction does not require a defined genetic mutation to occur. Aging, oxidative stress, environmental exposure, and chronic inflammation can impair oxidative phosphorylation gradually. ATP production decreases. Reactive oxygen species accumulate. Calcium buffering becomes unstable. These changes may not destroy neurons immediately, but they alter how effectively neurons fire, maintain synaptic strength, and repair structural damage. Axons are particularly vulnerable to metabolic stress. They extend long distances and require distributed mitochondrial populations to sustain ionic gradients along their membranes. When ATP supply becomes marginal, sodium-potassium pumps operate less efficiently. Membrane potentials fluctuate. Conduction becomes unreliable. Signal timing shifts subtly. These changes do not appear on MRI. They appear as cognitive slowing and motor inefficiency. Neurotransmitter recycling requires energy. Vesicle transport requires intact cytoskeletal machinery powered by ATP. When mitochondrial output falls, synaptic transmission becomes inconsistent. Communication between neurons weakens before neurons die. The network becomes noisier and less precise. Calcium dysregulation amplifies the problem. Mitochondria normally buffer intracellular calcium, preventing excitotoxicity. When buffering capacity declines, transient spikes in calcium become more damaging. Enzymes that degrade proteins and membranes activate inappropriately. Microstructural damage accumulates slowly across thousands of synapses. Hippocampal volume appears preserved, yet performance deteriorates because metabolic resilience is reduced. Tasks that require sustained activation become exhausting. Processing speed slows because neuronal firing rates are limited by energy availability. Oxidative stress compounds mitochondrial vulnerability. Reactive oxygen species damage lipids within myelin, proteins within synapses, and DNA within mitochondria themselves. Repair mechanisms attempt compensation, but over time, cumulative oxidative injury shifts the balance toward dysfunction. The process is diffuse rather than localized. Importantly, metabolic decline can exist without inflammatory markers or visible atrophy. Standard laboratory tests do not measure mitochondrial efficiency within cortical neurons. Cerebrospinal fluid biomarkers do not capture subtle ATP deficits. Before that threshold, the dysfunction is below detection. Energy collapse precedes structural collapse. As ATP deficits persist, axonal transport slows further. Mitochondria themselves become fragmented. Cytochrome release pathways activate. Apoptotic signaling increases. Only after prolonged metabolic stress do neurons undergo irreversible degeneration. The clinical presentation of mitochondrial inefficiency overlaps with psychiatric and functional diagnoses. Fatigue, slowed cognition, difficulty sustaining attention. These symptoms can be misinterpreted when imaging appears normal. Yet beneath that normal scan, synaptic networks are operating under metabolic constraint. Neurodegeneration without diagnosis may therefore arise from energy failure before cell death. The nervous system can appear structurally intact while losing efficiency at the molecular level. White matter conduction slows not because tracks are severed, but because ATP is insufficient to sustain optimal signaling. Synapses weaken not because plaques are present, but because vesicle cycling falters. This stage represents a transitional vulnerability. If metabolic stress continues unchecked, axonal degeneration will follow. But before that threshold, the disease exists in a gray zone, functional impairment without structural confirmation. The brain is alive, yet underpowered. The absence of a lesion conceals a system already operating at reduced capacity, one sustained on diminishing energy reserves without a name to define it. Network disintegration, the brain as a timing system. The brain does not function as isolated regions performing independent tasks. It functions as a timing system. Every complex behavior, speaking, planning, walking, deciding, depends on synchronized activation across distributed circuits. Signals must arrive within narrow temporal windows to produce coordinated output. When timing degrades, integration fails even if neurons remain structurally present. White matter inefficiency, subthreshold proteinopathy, and mitochondrial stress converge at this level. Conduction slows unevenly across tracts. Synaptic transmission becomes inconsistent. The result may be progressive desynchronization across distributed networks. Neural networks lose coherence without undergoing visible destruction. Frontal subcortical circuits are particularly sensitive to timing disruption. These circuits connect prefrontal cortex to basal ganglia and thalamus through long myelinated pathways. They regulate initiation, inhibition, sequencing, and cognitive flexibility. If conduction delays increase within these loops, task initiation becomes unreliable. The individual knows what must be done, but struggles to activate the sequence efficiently. Callosal fibers coordinating interhemispheric communication also degrade in timing precision. Tasks requiring bilateral integration slow. Multi-step reasoning becomes fragmented. Executive function deteriorates not because of memory loss, but because communication between cortical regions is mistimed. The cerebellum provides another example. It refines movement through rapid feedback loops that depend on millisecond-level precision. Even slight conduction delay can destabilize coordination. Gait becomes inefficient. Turning requires conscious correction. These changes may appear subtle and inconsistent, yet they reflect measurable desynchronization at the network level. Desynchronization is not equivalent to neuronal death. Neurons continue to fire, but their outputs arrive out of phase. When excitatory and inhibitory signals are mistimed, circuits expend more energy correcting noise. The brain compensates by increasing conscious effort. Tasks that were once automatic now require deliberate control. Fatigue increases because compensation consumes metabolic reserve. Functional imaging studies in similar states often reveal reduced connectivity between key hubs rather than focal lesions. The default mode network, executive control network, and salience network may show decreased coherence. However, these findings require specialized analysis and are not part of routine diagnostic evaluation. Network disintegration also explains why symptoms may fluctuate. Variability in metabolic state sleep quality, or minor stressors can further alter timing precision. On some days, synchronization improves marginally. On others, it degrades. This variability complicates diagnosis because decline is not uniform or catastrophic. Importantly, timing failure can exist for years before axonal degeneration becomes evident. The brain may operate in a desynchronized state long before structural collapse is visible on imaging. During this period, inefficiency accumulates. The patient senses decline, but no defined syndrome captures it. When networks fragment beyond compensation, structural degeneration follows. Axonal stress increases as circuits attempt to fire repeatedly to overcome noise. Energy demand rises. Calcium dysregulation intensifies. Eventually, axonal continuity is compromised. The transition from desynchronization to disconnection marks a critical threshold. Neurodegeneration without diagnosis often lives in this prolonged phase of network instability. The architecture appears intact, yet integration fails. The nervous system remains conscious, articulate, and present, but its timing infrastructure erodes. Without synchronized communication, even intact neurons cannot generate coherent behavior. The brain is not merely a collection of cells. It is a dynamic system dependent on temporal precision. When that precision dissolves gradually across networks, function declines without focal destruction. This is the silent progression of unclassified brain failure. Not sudden collapse, but erosion of synchronization beneath the threshold of recognition. Why imaging looks normal, structural visibility versus functional collapse. Modern neuroimaging is powerful, but it is not omniscient. I can detect tumors, hemorrhage, infarction, and significant atrophy with remarkable clarity. It can visualize demyelinating plaques and measure ventricular enlargement. Yet its resolution is limited. Each voxel represents millions of neurons, glial cells, synapses, and axonal segments. Microstructural degeneration, distributed evenly across those voxels, may not alter signal intensity enough to appear abnormal. Diffuse pathology is particularly difficult to detect. Imaging algorithms are optimized to identify contrast, differences between damaged and preserved tissue. When degradation is symmetric and gradual, contrast diminishes. The entire system shifts subtly rather than one region collapsing distinctly. Radiological interpretation may therefore describe findings as within normal limits or nonspecific. White matter microstructural damage can exist without producing hyperintense lesions. Myelin thinning, axonal metabolic stress, and low-grade gliosis may not create discrete boundaries visible on standard sequences. Diffusion tensor imaging can sometimes reveal fractional anisotropy changes that reflect reduced tract integrity, but such analysis is not routinely performed in standard clinical practice. Similarly, cortical thinning must reach a measurable threshold before volumetric analysis identifies atrophy. Early neuronal shrinkage and synaptic loss may reduce functional output without altering growth structure. Functional decline can precede visible volume loss by years. Positron emission tomography and other advanced imaging modalities detect metabolic changes or protein deposition, but they rely on density thresholds. Subthreshold proteinopathy or mild hypometabolism may not meet criteria for abnormality. Negative imaging does not exclude ongoing cellular stress. Inflammatory markers further illustrate this limitation. Cerebrospinal fluid analysis can detect overt pleocytosis or elevated protein, but low-grade microglial activation may not alter measurable parameters. Neuroinflammation can persist below detection thresholds while contributing to chronic degeneration. The expectation that pathology must be visible to be real creates diagnostic reassurance where none is warranted. A structurally normal MRI can coexist with significant conduction inefficiency, synaptic weakening, and network desynchronization. The imaging appears stable because macroarchitecture is preserved, even as microarchitecture erodes. This gap between structural visibility and functional collapse is central to unclassified neurodegeneration. Medicine relies heavily on imaging to validate symptoms. When scans appear normal, alternative explanations are sought: psychiatric framing, stress-related dysfunction, or functional neurological disorder. Yet imaging reflects anatomy at a coarse scale. It does not measure timing precision, metabolic resilience, or synaptic integrity directly. Over time, if degeneration continues, imaging will eventually reveal change. White matter may thin, ventricles may enlarge, cortical atrophy may become measurable. But by the time these changes appear clearly, the disease has advanced beyond its silent phase. The patient inhabits the interval before that visibility. Symptoms accumulate, processing speed slows, executive control falters. Yet repeated scans show no dramatic progression. The absence of radiographic change can reinforce diagnostic uncertainty, prolonging the gap between functional decline and formal recognition. Neurodegeneration without diagnosis persists because our tools detect only certain scales of damage. Structural collapse must cross a boundary before imaging confirms it. Below that boundary, degeneration is real but radiographically subtle. The brain can be failing while appearing intact. Imaging normality does not equal biological stability. It reflects the limits of resolution and contrast detection. In the space between cellular dysfunction and macroscopic lesion, functional collapse can proceed quietly, unvalidated by scans, but grounded in structural processes beyond the reach of routine observation. The point of no return. When axons begin to die, the nervous system may operate in a state of inefficiency, slowed conduction, metabolic strain, desynchronized networks, without irreversible structural loss. But there is a threshold beyond which compensation fails permanently. That threshold is axonal degeneration. When axons begin to fragment and disconnect, the process transitions from functional instability to structural absence. Demyelination and metabolic stress increase the energetic cost of conduction. Sodium channels redistribute along exposed segments of axonal membrane. ATP demand rises to maintain ionic gradients. If mitochondrial output cannot meet this demand, ionic imbalance develops. Sodium accumulates intracellularly. Calcium influx follows through reversed exchangers. Calcium-dependent enzymes activate and degrade cytoskeletal proteins that maintain axonal integrity. This cascade unfolds at a microscopic scale. The axon swells. Transport of proteins and mitochondria slows. Microtubules destabilize. Eventually, structural continuity is lost. Fragmentation begins distally and progresses proximally. Once an axon is severed, signal transmission ceases entirely. Unlike myelin, which can sometimes be partially restored, long central nervous system axons do not regenerate meaningfully across large distances. Developmental growth cues are absent. Inhibitory molecules dominate the extracellular environment. Astrocytic gliosis fills the space left by degeneration, forming a structural scar that stabilizes the region but prevents reconnection. The transition from desynchronization to disconnection is not always clinically dramatic. Symptoms may appear to worsen gradually. Tasks that were once inconsistent become consistently impaired. Initiation does not recover even on better days. Fatigue becomes constant rather than fluctuating. The variability that once characterized early decline narrows into permanence. Imaging may begin to reflect these changes as white matter thinning or subtle ventricular enlargement. However, even at this stage, findings may remain nonspecific. The structural loss may be diffuse rather than focal, lacking the signature pattern of a named disease. Axonal death also amplifies degeneration in neighboring fibers. Degenerating axons release signals that activate microglia. Inflammatory mediators increase oxidative stress in adjacent tissue. The environment becomes progressively hostile to survival. A self-reinforcing cycle emerges in which metabolic stress and structural loss accelerate one another. Network-level consequences become fixed. Frontal subcortical loops cannot reassemble once fibers are severed. Interhemispheric communication remains permanently degraded. Compensation strategies no longer restore function because the pathways required for integration no longer exist. This is the point of no return. Not because symptoms are severe, but because the substrate for recovery is gone. Even if metabolic conditions improve or protein accumulation slowed, the disconnected circuits cannot reconstruct themselves. The architecture that allowed synchronized activity has been dismantled. The fear embedded in this stage is biological, not psychological. The patient may remain aware, articulate, and cognitively intact in isolated domains. But the ability to translate intention into coordinated action has crossed into irreversibility. Each additional axonal loss compounds functional collapse. Neurodegeneration without diagnosis eventually converges here if progression continues. The absence of a name does not alter the trajectory. Structural disconnection defines the transition from inefficiency to permanent impairment. Once axonal continuity is lost, the nervous system cannot restore the networks that once sustained integrated human function. The disease may still lack classification. It may not meet criteria for Alzheimer's, Parkinson's, or any defined syndrome. Yet the structural reality is clear. Integration can dissolve beyond functional recovery. This reflects structural loss that is not currently reversible within adult central nervous system architecture. Functional collapse without a label. Dependence before diagnosis. By the time axonal loss becomes functionally decisive, the patient's daily life has already shifted in ways that are difficult to categorize. There is no definitive moment when a diagnosis is delivered. There is no single catastrophic event. Instead, independence erodes gradually, while medical classification remains unresolved. The individual enters a phase of functional collapse without a name. Executive failure becomes central. Tasks that require planning, sequencing, or sustained coordination cannot be completed reliably. The patient may begin activities, but fail to finish them. External reminders no longer compensate effectively because the internal networks required to translate cues into sustained action are structurally compromised. Effort remains present. Execution fails. Work performance deteriorates first in pace and reliability rather than competence. The person understands expectations, but cannot meet them consistently. Deadlines are missed. Multi-step responsibilities overwhelm integration capacity. Supervisors may interpret inconsistency as distraction or burnout. The underlying pathology remains invisible because insight and articulation are preserved. Social withdrawal often follows. Group conversations require rapid integration of auditory input, memory retrieval, and motor speech coordination. Desynchronized networks struggle under these demands. Response is slow. The individual avoids complex interactions to conserve energy. Withdrawal may be misread as mood disorder rather than neurological limitation. Motor function reflects similar patterns. Strength remains largely intact, yet coordination falters. Gait becomes cautious. Turning requires conscious adjustment. Dual tasking, walking while speaking, becomes unstable. Memory may remain intact in structured testing environments. This preservation complicates diagnosis further. Classic dementia patterns are absent. The patient remembers events, recognizes faces, and understands context. What fails is integration across systems required to act on that knowledge efficiently. The distinction between knowing and doing becomes pronounced. Because no diagnosis anchors the decline, misinterpretation persists. Psychiatric explanations may resurface. Functional neurological disorder may be considered. Stress may again be blamed. The patient senses progressive loss, but lacks a label that validates it. The absence of classification becomes an additional burden. Dependence emerges incrementally. Driving becomes unsafe due to slowed reaction time and coordination inefficiency. Financial management becomes unreliable. Household tasks require assistance. Responsibilities shift quietly from patient to caregiver. Fatigue intensifies. Cognitive effort required to perform basic activities increases as networks compensate for lost synchronization. The energy cost of functioning rises, while metabolic resilience declines. Rest does not restore prior capacity because the limitation is structural, not motivational. Awareness remains intact. The individual recognizes decline and anticipates further loss. This preservation of insight distinguishes unclassified neurodegeneration from many advanced cortical dementias. The mind remains present within a system that no longer executes reliably. Functional collapse without a label exposes a fundamental gap in medicine. Biological degeneration has progressed beyond compensation, yet classification systems have not caught up. Treatment pathways are unclear. Prognosis is uncertain. Support services may require a formal diagnosis that has not been assigned. Over time, the practical reality overrides diagnostic ambiguity. Assistance becomes continuous rather than intermittent. Structured environments replace autonomy. It merely obscures it. In advanced cases, structural disconnection may occur, leading to dependence even in the absence of formal classification. The label remains absent, but the structural failure is undeniable. This stage represents the lived consequence of unclassified brain failure. Biology has moved forward regardless of terminology. Stable aftermath. It is a limitation of classification. The nervous system can fail through diffuse white matter instability, subthreshold protein aggregation, mitochondrial decline. Biology does not require a diagnostic label to proceed towards structural collapse. The progression begins beneath thresholds. Myelin thins without forming plaques. Proteins misfold without forming dense inclusions. Mitochondria falter without triggering immediate cell death. Biomarkers remain within reference ranges. The patient senses decline long before the system acknowledges it. Axons fragment under metabolic strain. Gliosis replaces lost fibers. Circuits disconnect. Executive control falters permanently. Compensation strategies fail. Independence erodes. The brain remains conscious, aware, and articulate in isolated domains, yet the integration required for coordinated function is gone. Diagnosis depends on pattern density and reproducibility, not on the absence or presence of degeneration itself. The absence of a name does not imply absence of pathology. It reflects the boundaries of detection. Irreversibility reflects progressive structural loss within neural networks. Once gliotic scar stabilizes damaged tracts, meaningful long-distance regeneration is not known to occur in the adult central nervous system. The fear embedded in this condition is not rooted in uncertainty alone. It is rooted in awareness without explanation. The individual experiences progressive decline while existing between categories. The ambiguity delays validation, but does not delay degeneration. Neurodegeneration without diagnosis is therefore not the absence of disease. It is disease operating below or between established frameworks. It reveals that timing, metabolism, and connectivity can erode before lesions become visible or proteins become measurable. The stable aftermath is not recovery. It is permanence. The mind may persist, but the infrastructure that once supported effortless integration does not return. Biology has imposed its limit independent of classification. This is pathology of fear, not because the disease is unnamed, not because medicine has failed intentionally, but because the brain can deteriorate in ways that outrun our categories, crossing irreversible thresholds before recognition arrives. And this fear is real.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.